The cytoplasm of bacterial cells is where the functions for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, wastes, and gases, and contains cell structures such as ribosomes, a chromosome, and plasmids. All the other cellular components are scattered throughout the cytoplasm. Now, bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. Let's start with the cell wall, which is found in the outer surface of the cell membrane, and its most important role is to protect the bacteria. It consists of peptidoglycan, which usually helps differentiate whether the bacteria are gram-positive or gram-negative. Gram-positive bacteria have a single thick layer of peptidoglycan above the plasma membrane, which allows them to retain the staining dye. And gram-negative bacteria have a thinner layer of peptidoglycan sandwiched between the surface membrane and the plasma membrane, so they can't retain the dye. Additionally, the cell wall helps maintain their shape. Some species of bacteria are covered by a capsule made up of polysaccharides or complex carbohydrates, which act as a shield that protects the bacteria against phagocytosis and also help the bacteria adhere to surfaces. The capsule is a major virulence factor in the major disease-causing bacteria since the strains that lack a capsule are less virulent. Finally, the bacteria can move or attach to other cells and surfaces with the help of structures that can be found on their external surface. So, they have pili or pili, which are small hair-like projections emerging from the outside cell surface. These outgrowths assist the bacteria in attaching to other cells and surfaces such as teeth, intestines, and rocks. Without pili or pili, many disease-causing bacteria lose their ability to infect because they're unable to attach to host tissue. Another structure found in the cytoplasm is the nucleoid, which is an irregularly shaped region that contains the cell's chromosomes. The chromosomes of most bacteria are a single circle of double-stranded DNA, but some bacteria can have a linear chromosome, while others can have more than one chromosome. These chromosomes contain the genes needed to provide instructions for protein synthesis in the cell. Underneath the bacterial cell wall, there's a layer of phospholipids and proteins called the cytoplasmic membrane, which encloses the interior of the bacterium, regulating the flow of materials in and out of the cell. This is a structural trait bacteria share with all other living cells, a barrier that allows them to selectively interact with their environment. Membranes are highly organized and asymmetric, having two sides, each side with a different surface and different functions. Membranes are also dynamic, constantly adapting to different conditions. Now, in the bacterial cytoplasm, there are scattered cell components such as ribosomes, which are microscopic factories found in all cells, and act as a site of protein synthesis. These ribosomes can be found spread all over the cytoplasm, where they produce proteins that are destined to remain inside the cell, or they can be attached to the plasma membrane, and they are called plasma membrane-associated ribosomes. And in this case, they make proteins that will reside in the cell envelope or get transported outside the cell. Additionally to the genetic material contained within the nucleoid, many bacteria contain extra chromosomal DNA molecules called plasmids. These plasmids are small double-stranded DNA molecules that can exist independently of the chromosome and that can be circular or linear. Plasmids have relatively few genes, and their genetic information is not essential to the bacteria, and cells that lack them usually function normally. However, plasmids often contain genes that confer a selective advantage to the bacteria, such as resistance to antibiotics. Flagella, or in singular form, flagellum, are hair-like structures that provide a means of locomotion for those bacteria that have them. They can be found at either or both ends of a bacterium or all over its surface. The flagella beat in a propeller-like motion to help the bacterium move toward nutrients, away from toxic chemicals, or in the case of the photosynthetic cyanobacteria, toward the light.